Hello students, today we will be studying histology of GIT. In that we will be doing one important slide, that is of liver. Now liver is the largest gland of the body and weighs about 1.5 kgs. It is a modified exocrine gland. Anatomically, we talk of lobes of the liver, so liver can be divided into right, left, caudate and quadrate lobes. Now liver is supplied by hepatic artery, which gives 20% of the blood supply, and this is oxygenated blood. And the portal vein provides 80% of the blood supply, and this is the blood collected from the GIT. Liver is drained by hepatic veins, which ultimately drain the blood into the inferior vena cava. Now, when we go on to see the structural organization of the liver, the functional components here are capsule and connective tissue stroma, parenchyma, that is the hepatocytes, sinusoids and other blood vessels, perisinusoidal space of Dixit, and bile canaliculi. So we will be studying histology of liver under these headings. So let us first see the connective tissue capsule. Liver is enclosed by connective tissue capsule, which is called as Gleason's capsule. Connective tissue extends in the liver parenchyma along with branches of portal vein, hepatic artery and bile canaliculi. Reticular fibers, which are type 3 collagen fibers, form a network or a reticulum in the liver to support the parenchyma. We now go on to study the hepatocytes. Liver parenchyma consists of the liver cells called as hepatocytes. These are large polygonal cells which measure between 20 to 30 micrometers in size. Each cell having a large spherical nucleus at the center. Nucleus shows 1 to 2 nucleoli, and many hepatocytes may be binucleated. Cytoplasm is slightly acidophilic, that is eosinophilic, and granular. Lifespan of the hepatocytes is 5 months. Regeneration capability. Liver has capability of regeneration after loss of liver tissue. Now what we see here is a picture showing us the hepatocytes. So what we see here are the polygonal cells or hepatocytes, which are seen as cords or plates of cells. These are seen radiating away from the central vein, and they radiate towards the outer surface, where at few places we see these three structures, that is a bile duct, a tributary of the portal vein and a branch of the hepatic artery, and this together is what is called as the portal triad. Also what is seen here is in between the cords of hepatocytes we see the sinusoids. So when we go on to see the surfaces of these liver cells or surfaces of the hepatocytes, there are three surfaces which I have described. Sinusoidal surface faces adjoining sinusoids and shows microvilli that project into the space of Dixit. Microvilli increase the surface area for transport of nutrients across the hepatocytes. Second is the canalicular surface that forms the wall of bile canaliculars along with adjacent hepatocytes. And third is the intercellular surface that faces towards the adjacent hepatocytes. Again here a diagram showing us the hepatocytes, polygonal cells with a spherical nucleus in the center. Some cells also showing 2 nuclei. And what we see here is how these hepatocytes are arranged as cords or plates. And in between these cords or plates, we see the sinusoids. Next, we go on to study the liver lobules. There are three different types of lobules which have been described. First, we see the classical liver lobule. This is a hexagonal mass with central vein in the center surrounded by radiating anastomosing plates or cords of hepatocytes. Adjacent plates of hepatocytes are separated by sinusoids. A small quantity of connective tissue separates adjacent lobules. The outer angles of the lobule show presence of portal triad, and this triad is made up of branch of hepatic artery, branch of portal vein, and interlobular bile duct. This here is a diagram showing us the classical liver lobule. So as is seen here, each lobule is about polygonal or hexagonal in shape. The center shows presence of central vein, and from here we see radiating cords or plates of hepatocytes. And when we see the outer angles, the 6 outer angles, at each of these we see presence of a portal triad. So that is how a classical liver lobule looks like. At the portal triad, we see 3 structures. And how do we differentiate between these 3? When we see branch of hepatic artery, it's going to have a circular lumen and it's going to be lined by simple squamous epithelium. The next we see the branch of portal vein. This has an irregular lumen and luminal surface is lined by simple squamous epithelium. And the third structure there would be an interlobular bile duct, which again will have a circular lumen and will be lined by simple cuboidal or columnar epithelium. This here is a diagram showing us the portal triad. So we see the 3 structures here: the portal vein, bile duct, and branch of hepatic artery. Now, hepatic artery will have a circular lumen and a thick wall. It will be lined by simple squamous epithelium. The vein will be larger, having an irregular lumen or a collapsed lumen, and luminal surface lined by simple squamous epithelium. The third would be the interlobular duct, which is lined by simple cuboidal or simple columnar epithelium with a rounded nucleus. At the angles of this hexagonal classical liver lobule, the portal triad runs in portal canals or the connective tissue area. Now let us see what is the space of Moll or periportal space of Moll. This is the space between connective tissue stroma of portal canals and the outermost hepatocytes, and this is a site for lymph formation in the liver. Next we study the portal lobule. This is a triangular area of liver parenchyma around each portal triad. So here in the center lies the portal triad. It is considered to be the functional lobule as bile duct, which is present in the portal triad, lies at its center, whereas central veins lie at the 3 corners of the portal lobule. This here is a diagram showing us the portal lobule. So as we see, the portal lobule is a triangular structure. At its center lies the portal triad, and its 3 angles show presence of the central vein. Next we see the liver asanas. This is structural and functional unit of the liver, diamond shaped or elliptical in shape, with 2 axes: short axis between 2 closest portal triads and long axis between 2 closest central veins. And this asanas is described as having 3 zones. Zone 1, which lies near the portal triad, receives more oxygenated blood and is exposed to more amount of toxins. Zone 2, which lies between zone 1 and zone 3, and zone 3, which lies near the central veins, it receives less amount of oxygenated blood, hence it is more prone to hypoxic injuries. This here is a diagram showing us the liver asanas. So this here is the liver asanas, which is a diamond shaped or elliptical area having 2 axes. Short axis running between 2 closest portal triads and long axis running between two closest central veins. And we also see here the three zones. Zone 1, which is nearer to the portal triads, Zone 2 and Zone 3, which is nearer to the central veins. Liver sinusoids. Cords or plates of hepatocytes are separated by liver or hepatic sinusoids. The sinusoids are lined by discontinuous epithelium, which is endothelium, with discontinuous basal lamina, stellate sinusoidal macrophages, which are the Kupffer cells. Space of Dixit or perisinusoidal space. Sinusoidal epithelium is separated from the underlying hepatocytes by a small extracellular space called space of Dixit. Microvilli of hepatocytes project into the space of Dixit. This is the site for hematopoiesis in fetal life. Hepatic stellate cells or ETO cells are mesenchymal cells that lie in the perisinusoidal space of Dixit, and they are involved in storage of vitamin A and lipids. Now let us see the different cells which are seen in the liver tissue. We study them as the cell, their location and their major function. Hepatocytes. These are seen in the cords or plates, and their function is that they produce the bile, they detoxify the toxic chemicals, and they store glycogen. Kupffer cells. These are seen in the lining of the sinusoids. These are modified macrophages, hence they help in phagocytosis. ETO cells or hepatic stellate cells. These are seen in the perisinusoidal space of Dixit. Their function is storage of vitamin A and lipids. These become myofibroblasts in liver diseases like liver fibrosis. Next, we study the sinusoidal endothelium. This lines the sinusoids, and function is that the gap between these endothelial cells facilitates transport. Hematopoietic cells, which are seen only in fetal life, they are seen in the perisinusoidal space of Dixit, and they help in hematopoiesis. Next, we go on to see the intrahepatic biliary tree. This drains the bile secreted by the exocrine liver and conveys it to the extrahepatic biliary tree through the right and left hepatic ducts. The intrahepatic biliary tree consists of bile canaliculus. This is formed between adjacent surfaces of the hepatocytes. Next comes the intralobular or intrahepatic ductules or canals of Herring. These are lined by simple cuboidal epithelium. Next comes the interlobular ducts. These run through the interlobular connective tissue septa. They are lined by simple cuboidal or columnar epithelium and form a part of the portal triad. Next come the right and left hepatic ducts. Interlobular ducts join to form right and left hepatic ducts that later join to form the common hepatic duct outside the liver. The bile flows centripetally opposite to the flow of the blood in the sinusoids. Bile flows away from the central vein and towards the portal triad. So this here is the intrahepatic biliary tree. Hepatocytes, which secrete the bile, which flows through the canals of Herring or intrahepatic ductules, then into the intralobular bile ducts, then into the right and left hepatic ducts, and finally into the common hepatic duct. A diagram showing us the same. The canals of Herring, then the interlobular ducts, finally into the right and left hepatic ducts, which join to form the common hepatic duct. Let us now see the blood vessels of liver. Liver has dual blood supply through hepatic portal vein, which gives 80% of the blood supply, and hepatic artery, which gives 20%. The portal vein carries nutrients and toxic materials absorbed from the intestine, blood from the spleen and pancreas. Blood from the portal vein and hepatic artery enters into the hepatic sinusoids. So, as we said, blood from portal vein and hepatic artery enters into the hepatic sinusoids. From there, it goes on to the central vein, sublobular vein, then into the hepatic veins, and finally it is drained into the inferior vena cava. What is to be noted here is that the hepatic veins do not have valves. This now what we see is an H&E stained slide of the liver. This is the scanner view where we see the classical hepatic lobules having the central vein at the center and having portal triads at its angles. We also see the radiating cords of hepatocytes which are going from the central vein towards the portal triads. Another low power view showing us the slide of liver where we again see the central vein and then we see radiating cords of hepatocytes going towards the portal triad. A high power view of the slide of liver showing us central vein, hepatocytes as radiating cords or plates going towards the angles where we see presence of the portal triad. Another high power view showing us central vein and the portal triad having the three structures. We see a high power view showing us a portal triad where we see a large branch of the portal vein with an irregular lumen and a thin wall. We see a branch of the hepatic artery, thicker wall as compared to the vein, lined by simple squamous epithelium, and we see the interlobular duct, rounded lumen lined by simple cuboidal to columnar epithelium. So all three components of the portal triad are seen here: portal vein, hepatic artery and the bile duct. This here is an aperio image scope picture of histology of liver. We appreciate the classic liver lobule with central vein at the center and portal triads at the angles. Another high power view of the same image which shows us a central vein. We see radiating cords of hepatocytes. We appreciate that each hepatocyte is polygonal or hexagonal in shape with a rounded nucleus in the center. We also see a portal triad where we appreciate the branch of the portal vein, hepatic artery and the bile duct. The salient features for spotting for a slide of histology of liver would be presence of hepatic lobule with central vein, presence of radiating hepatic cord cells, presence of portal triad. Thus, in this video, we have studied an important slide, and that is histology of liver. For further reference, you can read Sontake Yogesh, Textbook of Human Histology by CBS Publishers and Distributors Private Limited, New Delhi, 2020, page numbers 223 to 228. Thank you.